It's very, very, very difficult to measure the absolute abundance of carbon-13 and carbon-12. And so what we typically measure is we measure the ratio of those, the heavy to the light isotope. And not only that, we have to measure the ratio relative to something else. So just like if you have a balance and you're weighing something compared to a standard weight, you may not, it may take you, you have to spend a lot of money to buy the standard weight because it's a lot more difficult to determine its exact accurate value. But once you have it, you can determine the relative weight very, very sensitively with this kind of balance, because you can, if it's heavier or lighter than your standard weight, even though you don't know the absolute, you only know the absolute weight through the standard. And that's what we do with all isotopes, including radiocarbon and C13. So we have internationally accepted standard materials, and for carbon-13, that's this PD belemnite. It's a fossil that, someone carved out of a rock and said, this is going to be my standard. This is the creature that it looks like. They lived in the Cretaceous, so they're extinct now. And here are the values if you ever need them for that. So then the way you express things, because the differences between the ratio of your sample to your standard is often rather small, we basically are subtracting 1 and multiplying times 1,000 to get a number that's, in kind of a whole number unit. Whoops. There we go, that was the one I wanted. So if we look at the C13 values of the major carbon cycle reservoirs, if we go back to this, if we have the same value in a sample as our standard, then this would be 1 minus 1 times 1,000, so it would be 0 per mil. And so if we look at the ocean, since our standard is a carbonate that was originally coming from marine bicarbonate and carbonate precipitation of calcium carbonate, we expect values in the ocean of dissolved inorganic carbon to be close to our standard. So, and that's, you can see that's true. These are the delta C13 values for marine DIC and marine carbonates. That's about this. But if we look at organic carbon, so something that an organism has turned from inorganic to organic carbon, then you can see that we get these negative delta C13 values. So remember, a negative delta C13 value means that this ratio, the sample to the standard, has to be less than 1. And so then we get a negative number here. So if we had the ratio of the sample to the standard be 0.975, then we would get minus 25 per mil, which is a typical sort of pre-industrial C3 plant. And so, if we then have plant material that again gets fractionated, so that just means that the organisms are preferentially taking the C12 and making sugars out of it. And again, if we're making methane out of organic matter, the first things to react and the fastest reactions are going to be the bonds that you can break are going to be the C12 bonds. So we get these very, very depleted in C13 or relatively enriched in C12, things like methane emissions. So that's your 5-minute introduction to C13. What do things look like in a typical soil profile? So these are just some data that I have together with Beto Quesada from some of his thesis samples. These are just sites in terra firme tropical forests going from the eastern Amazon to the western Amazon. And what you can see is the typical depth profile of high organic carbon at the surface, declining rapidly, and then leveling off at depth. in these very deep soils. If we look at the carbon-13, what we see are values of about -28 per mil at the surface. And then you'll see this in many, many soil profiles that are, there'll be an enrichment of about 2 per mil as you go deeper into the soil, and then that kind of also levels off. Or in some cases, like this Zafiri soil, or even Tambopata, it seems to have a minimum and then enrich again as you go deeper. So one thing you can say is that the delta C13, this is of organic matter, so it's already something that was fixed from the atmosphere by biology, so that's why we have values that are depleted in C13 compared to our carbonate standard. Then we can start to ask ourselves, what's the cause of this increase in C13? As we also are losing a lot of carbon as we go deeper into the soil profile. So on the next slide, I kind of go through, there have been sort of 3 possible reasons that have been given for these patterns that are seen in many, many soil profiles. The first is that we have to remember that we're in a time of global change, and the delta C13 of the atmosphere has been declining over time. So this is a plot of delta C13 from the year 1850 up until the present, taken from ice cores and then atmospheric observations for the more recent decades. And what you can see is that pre-industrial atmosphere was about -6.5 per mil in delta C13. So it's controlled sort of halfway between the ocean and exchange with the ocean and exchange with the terrestrial biosphere. But that over time, as CO2 in the atmosphere has increased, and especially as it's increasing rapidly since 1950, the source of that CO2 is largely fossil fuel combustion and release from deforestation and burning in the biosphere. So what you see is a decline of delta C13 over time because the CO2 that we're adding to the atmosphere is low in C13 compared to what's there. And you can see that the decline is about 2 per mil if we go up to about 2020 overall. So if the carbon at the surface here is much, much younger than the carbon deeper, then that must be at least some portion of the signal that we see, although probably it doesn't explain all of it. So the second big explanation for the increase in C13 with depth has to do with this, a lot of the carbon that's just being disappeared is more fresh plant material that maybe most of it's being respired and some is being retained in microbial biomass or sorbed onto mineral surfaces. And those are processes that can fractionate carbon, right? Decomposition, you're breaking bonds. And so you would tend to respire the carbon-12 CO2 faster than carbon-13. And so we expect a little bit of enrichment as you go from fresh plant material to microbial biomass. Then the question becomes, if I decomposed 100%, the fractionation wouldn't matter because, everything that was fresh plant would be back in the atmosphere. So you have to store enough of this, a large enough fraction to really see a big effect in the organic matter. But that's clearly also a reason, and people have used incubations and things like this to show that that this must be also a process that's important. And then the final process is this CO2 fixation that happens, especially in carbon-limited deeper soils that can increase C13. And I just wanted to that may be one of the reasons some of these are turning around, especially in poorly drained soils. Okay, now on to radiocarbon, which is the main topic here. So the difference between radiocarbon and stable isotopes is that stable isotopes are created and in the center of stars and never, then their values are fixed. They can move around in between reservoirs, but they'll always be the same amount of carbon 13 and 12 on Earth. Whereas radiocarbon is what's called a cosmogenic isotope. You need very, very high energies for nuclear reactions. And we get those high energies on Earth now only through cosmic rays that are high energy particles that are impacting our upper atmosphere, breaking apart molecules and atoms. And they also do this at the surface, but very little penetrates the atmosphere and gets to the surface. So these are, radiocarbon is one of several what are called cosmogenic nuclides. Here, you break a nucleus apart, and one of the neutrons in it will eventually have the right conditions to knock a proton out of a nitrogen-14 nucleus. And because it's lost a proton, it's no longer nitrogen, it becomes carbon. But it has the same mass because you've replaced a neutron for a proton. Once the carbon-14 is made, it gets oxidized to CO2. It can then enter the Earth's carbon cycle from the atmosphere largely. And ultimately, its fate is, because it's a radioactive element, it will spontaneously decay back to the nitrogen-14, but it takes a long time for that to happen. So then the half-life, which I'll explain in a minute, is about 5,700 years. So we have carbon-14 at all because it's constantly being produced and constantly being lost. And it just takes a while for it to decay. So we have it in measurable amounts on our planet, which is great for us. So just like stable isotopes, we report radiocarbon data as a ratio, this time of carbon-14 to 12 with respect to a standard with a known ratio. And I'll make these slides, I can make a PDF of these available to you so you'll have all these notes to go back to if you want. I just didn't figure out how to do it beforehand. But the idea is there's one extra thing. So we have this thing, the unit that's most commonly used in radiocarbon is this fraction modern. You can also multiply it times 100 and call it percent modern. And all that is the ratio compared to a standard. The standard is basically fixed so that its value is the same as pre-industrial wood. So we want the pre-industrial atmosphere to be our standard. And then the other special thing we do is because radiocarbon has a mass that's different than carbon-12, it will also have this mass-dependent partitioning within the environment. And so we want to get rid of that part and just look at the time part of the radiocarbon signal. So the way we do this is all radiocarbon data that you will get from a lab as fraction modern, they've all been corrected to a common delta C13 value, as if everything, all carbon in fraction modern has a delta C13 of -25. And the reason we do that is because, I showed you before the delta C13 of CO2 in air would be say -8 per mil. In a leaf, it's 20 per mil lighter. And so the fractionation of, the leaf photosynthesis is about 20 per mil. So if that, we know that the mass difference of C13 to C12 is 1, and the difference from carbon-14 to 12 is 2, so that we expect twice the fractionation by mass. So a 20 per mil difference in C13 would be a 40 per mil difference in radiocarbon. And if we turn this into radiocarbon years, which I'll get to in a second, it's about 330 years. So we would have, even though the leaf is the same, created from the CO2 and air while we watch it grow. If we didn't take out this mass-dependent fractionation, it would seem like the leaf was older than the air from which it was fixed. So this is the equation that we use to remove it. What we do is we measure the delta C13 of the sample as we measure it, and then we automatically correct it to this minus 25 per mil value. So any number that you get from a radiocarbon lab will have this already in it. So the most common way people are used to seeing radiocarbon data reported is as radiocarbon ages. And so I just want you to know it's not an actual age. So if we look at the idea behind radiocarbon dating is that if I have, say, a tree ring or a seed or something that I can grow and then isolate from the atmosphere so that it's not exchanging carbon anymore, then we start with this fraction modern of 1. So, and then the rate of radioactive decay is this law that most people get in high school chemistry. So it's the decay rate times the number of atoms gives you the radioactivity or the rate of loss of carbon-14 atoms with time. And if you do all the math, you can basically convince yourself that the radiocarbon age is related to this loss rate, the radioactive decay constant, and then the natural log of the number of atoms that you have at any given time divided by the atoms that you had at the beginning. So the easiest way to think of this is the exponential decline, and the half-life is just the time it takes to lose half of the atoms that you started with. So you go from a fraction modern of 1 to a fraction modern of 0.5 in 5,730 years, which is the half-life of radiocarbon. And 25% would give you 2 half-lives, or about 10,000 or 11,000 years, right? So that's the basis of radiocarbon dating. You don't know the age of your sample, but you assume that it started with a fraction modern of 1. And if I measure it now and it's got a fraction modern of 0.4, I should be able to say it's about 18,000 years old or whatever, based on this. And the assumptions are that the atmosphere has been constant pre-industrially through time, that it's a closed system. So if you're in a soil, you're constantly adding new carbon and removing carbon. So those are not closed system. This only works for something that you know hasn't been exchanging carbon with the atmosphere. And it also has this assumption that we know the radiocarbon half-life. And I don't want to get into all the complexities of that, but we basically, because these, especially the first and the third assumption, are not very good ones, we actually don't use a radiocarbon age as not an age until it's been calibrated. And I'll just maybe go through that very, very quickly. So if we use the fraction modern for the pre-industrial world, the atmosphere would have had a fraction modern of 1. And the red things here are just how much of the total carbon in the world is in the reservoir. But if you go to something like the deep ocean, the ratios are lower than 1, and that's because the time it takes to exchange carbon in the deep ocean with the atmosphere is, maybe several 1,000 years to several hundreds of years. And so it's had time to decay radioactively since the last time the deep ocean was in contact with the surface ocean and the atmosphere. Similarly, the surface ocean is not in equilibrium with the atmosphere because it's constantly mixing with water in the deep ocean, as well as having gas exchange with the atmosphere, and so on. So again, if we look at our soil profile, here's our same tropical forest. So here's the carbon decline with depth. And what you can see, here's the fraction modern. So we see at the surface, actually, we have a lot of values that are greater than 1 fraction modern, and I'll come back to that in a minute. But then you see a fairly, as you go further away from the surface, the fraction modern is declining. And so, you know, if you look, say, at 2 meters depth, I mean, here's 0.5 modern would be about here, right? So that's about 6,000 years old carbon in some of these soils. And so what we want to do when we're looking at things, these are just like the sort of mean carbon-14 age. We can use it to say, you know, the carbon at this depth is thousands of years old on average. And, but we can't really use radiocarbon to say this is 6,000-year-old carbon. We have to model it, and that's where Carlos's talk next week will come in. So I don't really have time to go through this, but you know, people have used the fact that you can have reservoirs where we know the age of something, then we can measure the radiocarbon change and reconstruct back in time what the carbon-14 of the atmosphere was. And the important thing here is just to say that as we go back in time, so here, this delta, so then we get another notation, which is basically it's the same as the delta C13 notation. It's the ratio to our standard -1. And here we have some correction for what's the known age of this sample. And what we really want to know here is what's so here, instead of the fraction modern being 1, our delta for 1950 is 0, because, you know, the fraction modern, this would be the fraction modern, -1 times 1,000 would be 0 for delta. And so what you see is as you go back, these are thousands of years. So this would be, you know, the start of the last ice age, most of this. That as you go back in time, there was more radiocarbon in the atmosphere than there has been in the later parts of the Holocene. And there's lots of interesting research into the reasons for that. But what it means in practice is that if you just used the Libby assumptions and calculated a C14 age for something 10,000 years old, you would actually measure something much younger, because there was more C14 in the atmosphere when it began, and so the actual age is less than what you would assume. So these are these calibration curves. What you can do is plot the radiocarbon age versus this line here is the basis of all these known age samples that have been measured by many, many labs over time. And I think, and then it's where your errors intersect this curve is what's telling you the calendar age that goes with the radiocarbon age that you measured. And so there's a new IntCal that just came out, IntCal 2020. These are always changing. So it's important to always go back and report the fraction modern as the most important thing. So there's also this important thing to know that if you get something that's just like 150 plus or minus 50 years old, the calibration curve has some problems in the most recent past. So we have very big problems for things that were fixed between the 1600s and the 1950s. We have very little specificity with radiocarbon in that period. So just really quickly, the other thing we use that Carlos will spend most of his time talking about, I think, is this fraction modern greater than 1. How can we have that? Well, that means that there's more radiocarbon in your sample than there was in the pre-industrial atmosphere. And we know that radiocarbon is also made by humans through the use of bombs. And this is yet another unit of delta C14, but think of the delta. So the 0 would be the pre-industrial atmosphere. 1,000 would be 2 minus 1 times 1,000. So this essentially means that we doubled the amount of radiocarbon in the atmosphere in a very short, and this is the atmospheric measurements from the northern hemisphere in red and the southern hemisphere, where you can see it took a little time to get the bomb products from one hemisphere to the other. And what you can see is this rapid increase. There was a moratorium, an end to the atmospheric testing, although there were further tests afterwards that were smaller. So it's in essence, it's like an isotope spike for the whole carbon cycle on the planet. And a lot of work has gone into where does that go? Oh, I think oh well, I don't have that one, sorry. But you know, the carbon-14 is disappearing here because it's mixing into other reservoirs like the ocean and the land, which is why we can use it. It's invasion into like soil organic matter to say something about how fast it's turning over. So we do have this other unit. Again, I'll send this to you. The delta C14 is subtly different from delta because we want to have an absolute standard in time, and I'm just going to skip through here. Basically, our standard, when we measure it, it has a lower value of C14 every year because the carbon-14 in it decays over time. So we just have to correct for that, and that's the difference between the delta and the delta C14. So here's a summary of all these ways of expressing radiocarbon, because that's one of the more confusing things. And then I know I'm going fast, but I just wanted to say, you know, for this normal radiocarbon, we can do up to about 50,000 years from about 300 years. And for the bomb carbon, we can look at carbon cycling on much faster timescales by following this spike. And and the ways we use radiocarbon, which I hope the other talks will go through, we don't do too much determining the age. You can use it as a source tracer if you look at different sources of respiration, for example. But for most of what the others will talk about, like Carlos and Claudia, I guess, soils are open systems that are continuously adding carbon. So they're adding carbon-14. And then what we're looking at is how that moves through the soil and comes back out through decomposition. And that's the slide I was looking for earlier. And Carlos will go through this in more detail, but if we want to model things, if you think about just a one pool model where you know, the carbon is well mixed inside, if I have a turnover time of 5 years, then basically the peak from the atmosphere will be delayed by 5 years if I have that as an input, and then I'm looking at the outputs. And basically the peak will get lower and more diluted and lagged over time. So we can kind of conceptualize what things look like. For longer turnover times, things will just now be reaching the peak or even still increasing if they are very slow. So there are very few things where you can model things as a one pool. This is fine roots from Sweden where you have different time points. And if you can fit that, that's what we want to do. And if I can just take 2 more minutes, these are just results from an elevation gradient in California, where as you go up in elevation, the mean annual temperature decreases. The precipitation does increase, so productivity goes up a little bit. But if we look at change in radiocarbon over time, this is just from the low density fraction of surface. One of the most powerful things you can do with this is look at changes over time, and that's a little bit of what Felipe's doing. You can see like the lowest elevation site has the highest C14 initially. It gets the most bomb carbon in the first years, and it's also declining very rapidly parallel to the atmospheric curve. So this is just a blow up of this. Whereas the coldest was low to begin with. It was, minus 50 per mil, and now it's still coming up. So, so in summary, I hope I've given you a little bit of an idea what kinds of things radiocarbon can be used for, and really, if you go away with nothing else, the radiocarbon, like the stable isotopes, undergoes mass-dependent fractionation. But the way we report the data as this fraction modern delta C14 or delta any of these things I showed you, it represents time only. So that was a very, very fast tour of radiocarbon. And hopefully there'll be questions. And as I said, Felipe, I can send a file and maybe it can get distributed through a Dropbox or something, because I know it's a lot of stuff to go through and a lot of formulas.
Yeah, I think as we progress through the webinar, people are gonna get the more of an idea how it's used.
Yeah.
So, and we also gonna have questions by the end, and we have everyone's email because everyone signed up for this, so we can send the information, no problem.
Okay, I'll send you a copy of the PDF.
Great, thank you, and I'll start my talk.
Let me introduce you first. Thank you, Susan, for the talk. We already have questions. We'll be attend at the end of the webinar, as I already said. So now we have the talk of Dr. Felipe Aburto, that it's called Soil Type Controls on Long-Term Carbon Cycling Stocks and Dynamics in Temperate Deciduous Forest and Plantations. Dr. Felipe is a full professor at the University of Concepcion, a soil scientist that has been focused his work and research on carbon and nutrient cycles on forest ecosystems. So thank you, Felipe.
Thank you, Oscar. Not full professor yet, I'm associate, but anyways, thank you. So, well, Oscar already introduced the title of my talk, and I'm gonna give you a very fast taste of what we're doing within this project, ForCARE, which is a continuation of a previous project I was granted by the national agency. And well, let me see, I cannot control. So my presentation is divided in a short background of what we're looking at, what is our experimental design, the main results we have obtained so far, some conclusions, and then at the end we have questions for Susan and I. So there is a land is being changed very dramatically through the human history. And we have forests as kind of this ultimate ecosystem that many people refer as this natural environment that many people go for healing, or we obtain also very important services and resources. But forests many times throughout the history have been changed into more intensified uses, mostly agriculture or pasture. And this is currently still happening. There are some countries, like in Chile, in which deforestation has kind of come to a stop. But in many other developing countries, this is still a very big problem. Sometimes these forests also change into other intensive uses that are plantation forests, that are an alternative to this forest into a more productive system for obtaining wood products or cellulose or fiber. And in Chile, there's been a change. This red colors the change of land use for plantations. And you can see that in this central south region is been changing, and it's been increasing. through the years. This is from 1985 to 2011. And you see this increment, especially in the coastal range, and more recently in the Andes Piedmont. And well, we really, the change that is happening as a result of this land use intensification hasn't been studied much in the world. You know, more recently, there's been more studies, but in reality, we know very little about this change. Most of the research has focused on the change to farmland, mostly agriculture or grazing. So there is, in Chile, though it's not as important as the change from cropland to plantations, there was a significant part of the country that changed from plantations, from natural forest to plantations, and sometimes from natural forest to cropland to plantations, which is probably the most of the surface. So why we think it might be important to check what's going on with the carbon with these plantations is because it's been reported that as you have a more intensive management, especially harvesting periods that are more intense than a natural recurring disturbance might occur in forests. These are data from eucalyptus stands in Australia. And you see that when you have, this is the biomass carbon stock, just in the biomass, no soil. You see as you have a logging period, a logging frequency more intense in this managed system, the carbon in the biomass never recovered to the point in which it will recover naturally. Yeah, it takes many, many years, obviously, to recover. So we have here a time frame, 200 years. So you see that the natural regrowth forest can reach levels of biomass carbon much higher than what a more intensively managed system does. And this have an impact on carbon. This is a review paper from 2017. And you see that the carbon in the soil, this is model data. You see that in almost all scenarios that people have modeled this throughout a long-term change, you see that the carbon start reducing. Yeah, there is a huge variability though throughout the models, and especially when the change is recent and you don't have many rotations, the change in carbon could be quite small. But through the years, as the system is logged more frequently, you might see a more important decline. So this is another review that looked at different changes from natural forests to different other more intense systems. And you see that for from forest to plantations, there's been a report change of about 17% carbon. Yeah, but also we have seen an opposite trend. When you change a cropland to a plantation, you can see a gain on carbon. Okay, so it's not all bad. It's not that the plantations are just bad compared to cropland. It's that when you change from a natural forest that is not logged to a logged system that is harvested more frequently, you're gonna start seeing a decline in both biomass carbon as a total biomass, all type of tissues, and also on carbon in the soil. So there's a change too, and we have today a better idea that we have different pools, and different carbon pools at different depths will be affected differently. So in this other review, we see that the overall losses of soil carbon from logging, just from logging, are around 11.2% of change. Yeah, the most of the losses occur on the organic horizons. However, there is also losses in the mineral soil. So the deeper soil layers haven't been studied much. We only have a few studies that look at depths deeper than 100 centimeters. From this review, they look at 925 points, but only 21 of those points were very deep soil layers. So we don't really know much what's going on at deeper layers. I have a problem here with the, sorry, with the screen with too many, well, now it's good. So another thing that might be interesting to look at is how different soils responded to this more intensified management. And this is the same review, but looking now by soil order. And there are some interesting differences. Most of the soil orders represent losses in overall carbon, soil carbon, around 20% after logging. However, there are some soils that have not shown a response, and these are the andesol soils, so volcanic origin ash derived soils. And this is very interesting, and we also want to look into that. So temperate forests, the ones that we're studying, have an important amount of carbon that is vulnerable to losses, and have been actually also very recently changed and altered. So it's an important fraction per year, at about 0.35% of the carbon is being lost per year, and also has a significant stock of carbon reservoir. So there are very important and significant systems to look at. Another thing that we also know about the soil and the way they respond and how vulnerable the carbon is, that there is a relationship with mineralogy. So soils that have mineralogy dominated by 2:1 minerals generally have a higher pool of carbon that they can store compared to 1:1 minerals. These are forest systems, and you see here the axis is double than in this case for 1:1 minerals, and you can see that you can have per mass proportion of fine soil particles a greater amount of carbon stored in these systems. So the questions we want to ask is, what is the overall relative change in soil organic carbon stocks and CNP stoichiometry as a result of this land use change or land use intensification? So what role does soil type, mineralogy play? And how deep are the changes that are occurring? And also an ultimate question is the end of my talk is, could C14 help us deciphering which carbon pools are changing, or how these carbon pools at different depths and in different soil types are affected? So the way we assess this is we pick 5 sites with pair plots that have the condition of natural forest and plantations forest. But these 5 sites are very special because they represent ranges of different soil types. Yeah, we have soils that are residual, 2 soils that are residual, a residual from granite, a residual from schist in the coastal ranges. And these soils are dominated in the case of the granite by mineralogies that are mostly 1:1 minerals, while in the residual schist is mostly 2:1 minerals. And we have a range of volcanic soils from very recent ash deposit soils as an antisol to an andesol and a more evolved alphy soil, soil from volcanic origin. So the mineralogies change from mostly amorphous, pseudomorphous to crystalline. These are the setups. So we have 2 adjacent plots, and we evaluate some forest dynamics, litter production, litter decomposition, root biomass, soil respiration, leaf area index, and leaching. And in the center of each one of these plots, we have our soil pits And in addition, we dug up 4 augers to a depth of 2.4 meters. We wanted to be very deep because as we saw from the previous research, there's only a few reports of deep carbon storage and cycling in forest system. Although these are, we know that forests and roots in trees can go very deep. So it's important to assess what's going on at deeper depths. So we look at CMP reservoirs, we look at mineralogies, at different soil properties in all these replicates, and we study the distribution of these elements, and as I said, some soil properties to see how are they changing too. So as mineralogy goes, as I said, the recent ash deposits is mostly amorphous. This is X-ray diffractograms, and we can see that the recent ash is mostly amorphous, and there's some traces of chlorite and cristobalite. So for the young ash, we see a change. The halloysite starts showing up. There's some gibbsite too, and goethite. But as I said earlier, these are mostly pseudomorphous allophane, mostly dominated soils. And the old ash, we start seeing much more of crystalline mineralogy, mostly metahalloysite, halloysite, and goethite, and some cristobalite. As we move to the other residual soils, what we see is that the residual granodite is dominant by kaolinite and gibbsite, as well as the schist, but we have a change that these are dominated by HIB, hydroxide interlayer vermiculite, while the residual from schist is dominated by chlorite and vermiculite intergrades. So selected dissolution shows that the young ash soil is the one that meet the requirement only in the first 50 centimeters for andisols. The recent ash soil we see in the oxalate to dithionate iron shows mostly amorphous iron oxides, and also in the young ash we see that is a celandic type of andesol. Something that we did not expect that we saw on most of the site, this is just an example. The change to plantations produce some shifts in mineralogy that we didn't see before. Though this has been reported before, it's something that we generally don't hear very frequently, especially when the changes aren't very, having been occurring for many years. So in this case, we have plantations that are about 40 years old, maybe second rotation. And we see a change and a disappearance of gibbsite on this site. Other sites show a change on the crystallinity of pseudomorphous or of more easy to alter minerals. But in this case, we see this gibbsite, and we're still looking into what are the processes that are behind these changes. There are changes, obviously, in cation proportions and CEC, pH, and other variables. So now if we look into what this talk is mostly about, let's see carbon stocks and stoichiometry. Here are the changes in carbon. We have that the sites are different between them, obviously. As soils are different, carbon in content on them is different too. So the carbon stocks are the highest in the andesol and the granite. The schist and old ash has intermediate values, and the lowest values are in the recent ash, which is most a sandy soil. When we compare plantations again native forests, we see that in the granite and recent ash are the only sites that we see a significant difference, mostly as a reduction of carbon. Then when we look at carbon distribution. We see that the change does not occur in all the sites the same. The granite and the young ash are the ones, I'm sorry, and the granite and the recent ash are the ones that show the greater difference. We see that in the granite, plantations on the red are almost always lower in carbon than the native forests, but only significantly at the deeper layers. Similar is the case with the recent ash. We actually see in the recent ash an increment, significant increment on the surface, but the significant decrease in the subsurface. We see some other difference in the other sites, but they're not significant. When we look to the stoichiometry, these ternary plots show us the relative change in carbon, nitrogen, and phosphorus in the different size, schist, granite, residuals, old ash, young ash, and recent ash, and throughout all the sites. And we see that the sites that show most of the difference are the granite and the recent ash, which are the same that we saw a change in carbon. This difference is, in the case of granite, is driven by the 3 elements, while in the recent ash, the main difference given by changes in nitrogen content, and in the young ash, there's also some difference, but not as important in carbon and phosphorus. The other sites and throughout the site, this is not possible to discriminate. So now if we look into dynamics, I'm gonna move very fast through this. I was trying to actually check the time. Okay, so we have a change in above ground biomass in these plots, and we see that in most of the sites, there's a large difference between the plantation and the native forest and above ground biomass, and as a result on the stocks of carbon, nitrogen, and phosphorus. These are liter production throughout 2 years of measurements, and we can see that in most of the sites, we have the plantations produce more liter than the native forests. And obviously there are changes in the way, since these are pine plantations versus the deciduous forest, there's a change on the dynamics of litter production too. When we look at roots, it's the opposite. And most of the roots, I mean, most of the sites show that the native forests produce more roots than the pine plantations, being significant in almost all sites. When we look at now at soil respiration, we see that there are also some changes in the behavior throughout the year, but what we see is that at least 2 sites, the soil efflux is much larger in the native forest than the pine plantation. Now, when we look at dissolved organic carbon, is the opposite. Now, plantations are losing more carbon through DOC than the native forests. When we do a balance of what are the standing biomass in native forests against plantations, we see that overall, there's a significant greater amount of carbon in the standing biomass in the native forest than in the plantation. There's not a significant difference in litter production. There is a significant, non-significant difference too in the organic horizon, and the soil overall throughout the sites do not show a significant difference on soil carbon. When we look at the balance between, the balance of inputs and outputs, I don't know what happened here. So these are carbon outputs. We see that there is a significant higher input of carbon in the native forest through roots and litter flow, but mostly explained by roots, and a non-significant difference between the native forest and plantation for the outputs. And if we do the whole balance, we see that carbon is greater on the native forest. So this picture is just a cartoon of showing the total pools of carbon. Here we can see that the native deciduous forest has much higher carbon than the plantations. And we also see that this is mostly explained by the difference in above ground biomass. In the soil, the difference are not significant as we saw earlier. However, where there is difference are in the balances. So the balance in the case of the native deciduous forest is always more positive. So there's always a greater gain, and the gain per megagrams per hectare per year is about 1.7, when in the plantation is 0.45. So now, if we start looking at radiocarbon to see if it can bring us any, shed any light about this. So these are bulk fraction modern C14, and of the different sites, the old volcanic, recent volcanic, residual granite, residual schist, and young volcanic. And we see that the plantations in red generally have bulk bulk carbon that is a little older than what we find in the native forests. However, there's not like significant difference with this. Here in the granite, I put the question mark because we're still missing some data. We don't know how this is going to behave. Now looking per site, comparing the bulk radiocarbon, the depth distribution, we see that in these residual soils and the old ash soils, all that have more crystalline mineralogy, we have a reduction of delta 14C. You see here that the plantations in red in all these sites are throughout most of the depths lower on value than the native forests. It's important to notice this change happened in a very short time frame. Like seeing that these are maximum of second rotation plantations, the shift in radiocarbon is quite significant in these three sites. In the young ash, we don't see much of a difference in the surface, but they start deviating in the opposite than the other sites, where the plantations have become younger carbon at deeper layers in the young ash soil. And the recent ash soil, we're still having some problems to running those samples because they're very low in carbon. So we're waiting on some data. So not enough data to make any conclusion about that. We also look at the respire carbon. So we did some incubation experiment, and Claudia and her lab received us and helped us, Claudia Simsek, to do and perform all this analysis in Irvine. And what we saw is that in most of the sites, the respiration on the native forest sites was higher than in the plantations, except for this granite soil, in which plantations respire more and a higher fraction of their carbon. The site that respire more was the recent ash soil, which also is relevant to highlight, since this is the site with the lowest clay content. This is a sandy soil and has the lowest carbon of all sites. So when we compare the bulk soil fraction motor to the respire fraction motor, we see that the respire CO2 has generally a higher fraction motor signatures than what we get in the bulk soil carbon compartment. We also see a trend, as Sue mentioned earlier, that as you become lower, move down in the soil profile, you see a decrease in fraction motor. This is also noticed here. There's a significant difference between the O and A horizons against the B and C horizons in the soil profile. This is also seen on the respire fraction here. There's a significant relation between the depth and the F value. So now if we look at the soil respire radiocarbon by site, we find something. I will refresh your memory moving back a little bit, that in this soils with crystalline mineralogy, we find that the plantations have in generally older bulk carbon at most depths than the native forests. And if we look at the respire, you'll see that the respire carbon is generally older in the crystalline mineralogy soils in the surface, and then it becomes younger in the subsurface. Well, in the young ash soil, we don't find much of a difference except at these depths, but they seem very similar. And in the recent ash soil, we find that they're very similar on the surface. Then the plantation forests become younger in the subsurface and older at deeper layers. So some main conclusions of what we are so far is that forest plantation significantly alters soil properties, including mineralogy in the sites that we have seen. But there is a significant difference on the response of the different soil types. So it's very important when we talk about these processes that soil type is really a major controlling on carbon cycling and the response of carbon cycling to this intensification of use. So land use change or land use intensification alters soil carbon and nutrient stoichiometry and the soil carbon storage as a result mostly of a reduction and change of the type of carbon and nutrient inputs into the system. The plantations in crystalline soils display an older soil organic carbon than native forests, but respire old soil organic carbon too, suggesting that there is a lower stabilization in plantations. Something is happening either because the process of stabilization change or simply because of the input change that makes these sites display an older signature in the plantations. Interestingly, the andisol, and this has been reported recently by other researchers, are not as responsive as the other sites. Their capacity to stabilize carbon is truly amazing, and this shows again that these sites are very resilient to this kind of change. I believe these findings are relevant as climate change mitigation plans involve forestation efforts using commercial exotic fast-growing species. So we can use some of the results we have and the results that other researchers have produced to include in this plant soil types. Soil types very important to figure out where is more appropriate to have this kind of management and where we're gonna have a better carbon sequestration potential. So that's my presentation. Thank you very much for your attention. And special thanks to all the team at the lab and to the collaborators and obviously to the National Agency for funding this research.
Well, thank you, Felipe, for your talk. Also, thank you, Susan. Now we'll have an open mic session for questions. I already see that questions are being put in the chat. So I will like, if you have questions, please specify if your questions are directed to Susan or to Felipe. Felipe, I don't know if you see the questions that are written.
I now see it. Yeah. So
There's a question by Sasha.
Sasha, this data on productive native stand plantations. I'm wondering whether the overall carbon balance on the two stand specific plantation and the carbon plastic.
Maybe just to jump in for a second, in the chat, if you can, I don't know if everybody can see the chat. Yeah, you can. I put a link to download the slides if you want to just click on that from my part, because I saw there were questions about that.
Yeah, there's maybe about the four stands. I think the question that Sasha has, if I understand correctly. The native sites are secondary stands. They're being affected by a similar process than the plantation site was. In terms of age, most of our native sites, the oldest trees are about 40 years old. So they were affected by some anthropic disturbance too, but they have recovered since then. The plantations, however, have been managed, and they are, some of them in second rotation. And in one of the sites, we have plantations that have not been managed because it was put there for erosion control purposes after a fire that affect the area, and was planted by the Forest Corporation in Chile. And that is about 40 years old plantation in that site that hasn't been more managed, but only one rotation. If that's the question Sasha has. And
maybe you can raise.
Oh yeah, there is another, yeah. There's also about what are accumulating faster, but I think maybe that question was made before I finished, because I showed those results, right? The native forests are, if we say it like the carbon balance is more positive in the native forest than the plantations.
I don't know if maybe some of our panelists have questions. We also can attend those. Carolina just raising hand.
Hello, thank you for both presentation, really nice. I have a question for Susan, but also for Felipe, because I was thinking in something that appeared in both presentation. My question is if can environmental stress condition alter isotopic fractionation in radiocarbon sample? I will develop a little maybe my question. It's because I work with microorganisms and certain carbon, and I saw that in extremophilic microorganisms, the stress condition, like for example, low oxygen or high water content, doing that these microorganisms absorb different than certain carbon. And I don't know if with the 14 carbon is the same, if there are a different absorption for the microbial biomass, and if there are a way of doing this correction, because I saw in the presentation of Felipe that in the profile you have a different respiration, but maybe it's because you have a condition of stress of the microbial biomass for absorb this carbon, for doing the mineralization, I don't know.
So that's a good question. So again, with the radiocarbon, we're correcting for this kind of fractionation effect. So like you might see something in the microbial stress that it takes up different kinds of carbon. But what you can do, I mean, so the radiocarbon, this gets into the use of radiocarbon as kind of like a source indicator. For example, if the microbes are accessing carbon of different ages as their food source, then, and they shift their food source, you can see that, and that's part of, so the incubation, the reason Felipe is doing those is to look at what are the microbes, carbon sources, what are their carbon substrates, what's the age, and does it shift when you maybe have the plantation versus the native forest. So, but you can also think about this, there's a lot of literature, for example, on drying and rewetting soils, that this, so one of my PhD students has just had some results looking at this, and we can show that the carbon-14 is changing with the drying and rewetting compared to a soil that you don't dry and re-wet. And so it means that the drying and re-wetting is allowing the microbes to access some older carbon pool that they're not normally accessing. And then you can hypothesize why that is. So you might also see a change in the carbon-13, but it might be for a different, it might be for the same reason that the substrate has a different carbon-13, but it could also be that the microbes are fractionating the carbon differently. So, but the carbon-14, because we correct for the C13, it's not a fractionation thing. It's just, my combination of substrates shifted. So, and I think that's, one reason to do the incubations is, that tells you about fast changes and, what the microbes are doing. So I thought it was really interesting that in the deeper soil, you see differences like between the lithologies and also between the plantation and native forest.
Okay, thank you.
Francisco.
Okay.
I wanted to say something for the attendees, is that you can raise your hands too. And actually I just saw that Carlos is connected, and I promoted here so he can be on the screen.
Mm-hmm.
Okay. Francisco.
Okay, thank you. Thank you very much for very nice presentation, both of you. So my question to Susan is more or less the same question that Carolina said. Well, how critical is the isotope discrimination by assuming that soil microorganisms are consuming at the same time of label and native carbon? I mean, you know that there's some sort of discrimination. How critical is to assume that the microbes can eat, can consume the same velocity, I mean the same speed as in over time, the labeled carbon with respect to a native one? Should be, I think this is a point that is not much treated in the literature.
Yeah. So again, it's not really fractionation. It's telling you which source is more abundant. So that like the labile carbon would have, in theory, higher carbon-14, more bomb carbon-14, and the old carbon sources would have lower carbon-14. So I don't have a good slide here, but one of the key things is that you just take a soil and you incubate it. And Felipe showed this in one of his slides really fast. Like you look at the bulk carbon and then you look at the carbon that's respired, and the respired carbon is usually higher in C14 than the bulk that it's. So it means that there's, when Carlos comes along next week, we need at least these labile and older carbon pools, because the microbes are preferentially eating the younger carbon, and that's why the CO2 in the incubation is higher than what's left over. So yeah, I mean, I think there's a lot to explore there. There's not a lot of data like these with the incubations with C14 to look at this, but it's very, very clear that as you go deeper in the soil, the mean age of the carbon being eaten by microbes increases. So it may just be that there's less of this very young carbon to eat when you get deeper in the soil. So, as if you look at dissolved organic carbon, you see the same sort of pattern as you go deeper in the soil. The carbon-14 of the DOC also decreases with depth. And often the DOC and the microbially accessible carbon are the same in terms of radiocarbon. But there are very, very few studies that compare all these things.
Okay, thank you. It's wrong to say fractionation.
No. Well, it's fractionation implies that it's separated because of the mass of the isotope to me. But you can also say the microbes are selective in what they eat, right? Which is another kind of fractionation. So,
yes, kind of fractionation.
Yeah, I wanted to point out, right, that in this case, what we see in the soils is like, since the plantations were all established after the bomb testing, all of the carbon that they're inputting into the system, including the native forest too, because it also has a disturbance before. So all this new carbon, what is entering the system. So if you see that the plantations are getting older in carbon, that means that the microbes are eating everything they have, right? So, so the carbon that is entering the system is cycling fast and is being lost more. And what I pointed before is we have a different type of input. As native forests produce more roots, the plantations produce more litter. And the main source or loss of carbon in the plantation, together obviously with the respiration, is DOC is much higher, DOC production. So dissolved losses seem to be more important as carbon is mineralized in the surface and being leached down in the profile. So what the microbes are eating down there is different. It's a different kind of food. So that can explain some of those changes.
Yeah, the differences in DOC are really striking in your balances. And also that was one of my questions is, are the rooting depths the same for these pine plantations and the native vegetation? Are you partly losing carbon just because you have fewer inputs now in those deeper layers?
Yeah, that's, I mean, roots are much higher density when you look at the profile in the native forest at all depths. But production, we only measure it in the surface, obviously for technical reasons, was too hard to sample down, but only for the first 40 centimeters. So we have that annual root production is much higher. And that it can be explained too, because these are monoculture, monospecific plantations. So obviously the distribution of roots is different at the landscape level, right? Because they're planted in lines and they will have a different distribution spatially. So that could also explain our results. And the other ones are more diverse and diverse roots too, as a result of the plant diversity. Yeah, Claudia.
In this change in mineralogy, and I was wondering if you can talk about some of the other aspects. So it's been shown that fires can lead to changes in mineralogy, or it's been indicated. So I was wondering if there's a big change in the pH of the litter, and if the site that has the mineralogy change is the one that burned or not.
Yeah, so that site, it has some evidence of fire. I don't know if there are differences between sites in terms of fire. I imagine the sites in the coastal ranges were more affected by fire historically, also for clearing the land. But we have evidence too in, for example, the Andes soil there's in the Piedmont. There's some charcoal in the soil profile too. So that was also probably cleared by fire when they open it up for agricultural purposes, probably at the beginning. the site history, and that's a problem. Sometimes when you get back in time, some of the records are not there.
That's okay. And what about the pH of the?
The pH, yes, the pH changes. It tends to be lower on the plantation sites. They're not huge changes, but since it's logarithmic, you can, small changes can have a huge impact. We also saw a change in aluminum. The dissolved aluminum is higher, and the plantations in some of the sites, I repeat that again, very site specific. And that site specifically is the one that shows more strong changes in aluminum. Also, the andesol changed quite dramatically in terms of aluminum and silicon and oxalate extraction. So that site also have that kind of change that could explain some of these shifts in mineralogy, especially the reduction in gibbsite on the granite soil.
Professor Rafael.
Well, I think I'm not sure if this is a whole question, but I always, when I look at comparisons from native forests and plantations, I always set up my mind in a challenge that what we are comparing, if we are really comparing the same conditions for the soils, regardless that we can think that we are comparing that, I always wonder about it. And my question for Susan, maybe, or any other of the panel, is regarded to, is there any way that we can use isotope signals based on what I saw from Felipe's presentation and other components, that we could be a little bit more sure if we are comparing exactly the same baseline for making this sort of question about what will be the final effect of the plantations, if we are seeing effects of plantation, because there are so many things that are going on here. I mean, you have exudates coming from the plantations compared with the native forests are different. Probably all root systems that have been died before that we are comparing with what's going on with microbes activity. And so I always kind of like challenge myself. And I've seen the results from these sort of studies in the US sometimes that go completely in a different direction. And so, but always the major alignment goes through the soil type. That is something that actually coincides with what Felipe found, that the strongest effects are on soil type. So, but still, I wonder when you have some sites that have lost their A horizons, and then you are comparing some that probably do not have the same profile conditions. Is there a way for the isotopic signatures to tell us better, or to sort of like know that we are starting from that?
I mean, the only thing you can do is, you can't go back in time and sample before it became a plantation. That would be what you would have to do, right?
Okay. A really long
things, because what we're saying is they were the same, beforehand, and they've evolved differently because of the treatment. In some places, other places we do have that. I can't remember if you have some, Felipe, some archived soils, but probably not from the 1950s. But we have another project in Uruguay then where they're looking at a similar question. They have grasslands that have been afforested instead of native forests. But there they actually do have the sampling, at least of a grassland before, and then adjacent, a place that's been afforested. But I think the only thing you can do with this is look for patterns across lots of places, the way Felipe is doing. I mean, you have to have enough confidence that you can replicate the result in multiple places, and that can give you confidence that the vegetation effect has an overprint. And the complication here is that you have the vegetation overprint on things that are different to begin with because of the differing mineralogies. So it's a complex story to tell. I don't know, Felipe, you probably have a better answer.
Well, I think it's very difficult to find perfect sites. What we tried to do here was just trying to find soils that like are the same or very similar. We know the soil variability is huge. So we cannot, I mean, there's not soil profile equal to another soil profile. I mean, any pedologist can tell you that, right? It's like they could be side by side, but they're never gonna be the same, exactly the same. But they're similar enough that allow comparison, especially if they're close together and they're affected by the same type of environmental conditions. And that's what we try to do here, is just not picking sites that were too far away. I mean, for example, the granite site is only 30, 40 meters away from one pit to the next. So with that, we can ensure that are very similar. But not equal. And I think that's the problem anywhere, really. It doesn't matter what you do, unless you pick a site and you deforest it yourself and then plant it and wait 40 years and, and see if that's gonna happen. But I don't know if I wanna wait 40 years for answering this question.
But I guess what you're saying is the real thing that probably we need to. I mean,
Yeah.
really long-term studies, and we should be looking for that kind of things to happen at some point,
Mm-hmm.
because those are really strong basis scientific comparisons that we can sometimes really pursue. And I do think that we don't much on that. But I know Claudia's having another comment.
No, I think it's a great idea to establish some long-term plots, and this is a call to the audience for the next generation scientists to get involved. But there's a lot of networks that have explored this idea of long-term plots. There was one question from the audience on forest age and mineralogy, and I think Felipe mentioned that the forests are about the same age. So it's that there's probably not an impact, but oh God. Oh yeah, so I really thought that Rafael's comments was interesting on the fact that you might have lost part of the A horizon, especially given the terrain that you're working in, the relief. So sometimes you can line up some of these soil properties, and it might be worth plotting the data not from 0, but from some other depths if you think you lost the top. In the Arctic, that works. Like sometimes in some organic soils, and they have a lot of ice content, we can actually use radiocarbon to see that the soil has collapsed by the subsided treatment that I have, and the control is just offset, and they look exactly the same, just at the wrong depth. So it's a combination of kind of ice loss in mineral soil that might not work with radiocarbon, but maybe some of the other indicators of what would define a horizon, like the iron content or something, would line up if you play around with that.
Yeah, yeah, that's a good point. There is a paper that used the carbon isotopes of just C13 to try to find erosion evidence, but in this case, my main problem, and I think for everyone working with land use change, is you don't know if the change you're seeing is because of the same treatment, right? But because as a result of the land use change. So in here, I think it's the way I think it is. We just assume that the system is kind of the basic assumptions you have to make to be able to compare. Because if I start looking, for example, of iron content, it could be related, but it could also be changed, as those minerals can be altered too as a result of this treatment.
Can you use some of those like theoretically immobile elements like titanium and try to do some ratioing to that?
In the residual soils, I think that's possible. Not on the sedimentary, I mean the ash deposit, since those signatures are totally altered. But in the two residuals, we can do that. And those are probably, and especially the granitic site, is the one that is affected most by erosion. So of all sites we have, high slope those materials are not very cohesive, so they can be eroded very easily. So the result on that side, it could be partially explained by erosion. But I think the question is also, isn't that part of the treatment too? As you can also wonder that, right? If it's eroding faster, then it's also part of the effect of the management. So I don't know if erasing that signature or not. There's
Just another question.
Yes.
Yeah, go ahead. No problem.
Felipe, thank you. Did you you show in your slide that the amount of input, biomass input, was higher in the plantation forest compared with the native forest? Is that right? Or
no. Total above ground biomass was higher on the native forest. What was higher on the plantation is the litter-fall.
Litter-fall.
Yeah.
And yes, and how do you think that it influenced your final balance, carbon balance? Because you get more in native, of course, but also increasing amount of carbon in plantation. Have you considered the priming effect? Because this is kind of things that in many models are not present, and it means an extra mineralization of carbon. Even when you are applying carbon, fresh carbon, you are respiring more native carbon, and then your total balance is negative or neutral. Most of the modeling in climate modeling that include some soil modeling, like a Century model or Roth carbon model, claim that at the end of maybe 50 years or 100 years, you end up with a low carbon sequestration in the terrestrial ecosystems. Well, so some comments on that, I mean.
Yeah, I think priming could be playing a role in here. There is one of the correlations that are interesting is between carbon-nitrogen ratios and respired carbon. So we see that carbon-nitrogen ratio and carbon-phosphorus ratio kind of are the ones that have the highest correlation with carbon, the respired signature of carbon, the fraction motor of carbon. So that's kind of interesting and telling us something about also quality of the material that is coming in is probably different. We found that the litter in the plantation is higher in nitrogen and phosphorus than the native forest input as litter, since it's a deciduous tree, right? And the other one are evergreen. So there is that difference, and it seems that the microbes are happily eating the material, the litter material that comes from the pine, and that's not is not the problem. I think the problem is, I don't know yet, but I think the main problem is the changes in magnitude and the source. Roots are probably more edible than as a material than the litter. So probably the microbes in the native forest are happier to eat those root materials, fine roots decomposing, than the litter on top of the soil that is not incorporated. So I think that's the main problem, and as a result of that, you start seeing the shift on age or the relative age of the carbon in the plantation, as they are eating everything they have in the plantation, and there's not much new carbon coming in that actually is sequestered. But I think priming is, I think in every system is playing a role, right?
Playing a role. Yeah. Thank you.
One thing I want to mention that I think it's important to keep in mind that, because for someone it might be surprising that how actually it's looking like a balance of carbon is completely different than a cumulative amount of carbon that we found in the system. So eventually, if we analyze the annual balance of carbon, I guess that the numbers will shift for larger balance for the plantation. That's something that in the balance, I mean, you are not accounting for what the stock of the current stock of the carbon is. It's mainly what is the annual flux of carbon. And that's something that probably is just to clarify that point, that I guess is a little bit different from what it could be in the end.
Yeah, I think if you measure it by the tree, I think what Rafael is, if I understand correctly, right, is if you measure the tree sequestering carbon, is obviously higher on the plantation, as in compared to the native species.
Yeah,
But
I mean the carbon per hectare per year. I mean the flux from one year to the next. That's something that probably in the calculation, when you look at those numbers, are a little bit different than comparing the whole stock, the current annual stock.
Mm-hmm. Yeah, I think it could be like that. In this case, we only have, you know, this is our third year of monitoring, so we don't have long-term records, and we're only measuring the input as litter and roots, not any other material. So if a whole tree falls down, it's not part of our litter quantification, you know, like coarse woody debris is not incorporated, and coarse roots are neither incorporated. So only fine to medium roots and litter, mostly as fine woody debris and leaves. So it's not a whole system balance.
Mm-hmm.
I don't see any more questions in the chat. I don't know if the panelists.
Yeah, I think we're past the time already, no?
Yeah, some minutes passed, but I don't know if there is any question, maybe to close.
Yeah, before we finish, I would like to invite everyone that has joined us today to come back next week. We have a very interesting talks, and I hope that you can join us in every week webinar. And feel free to contact us, everyone, and try to maybe answer some of this question. Maybe what Rafael was saying, let's start a long-term experiment now that we have some base data, and maybe when we're older, we'll figure out if we were right today.
That's the great thing about radiocarbon, is you can keep measuring through your whole career.
Yeah.
Yeah, that's great. It's hard to get funding anyway.
Yeah, that's the only problem.
Yeah, I guess so.
Okay, thank you everyone very much for joining today. Special thanks to Sue for presenting and clearing up the idea of radiocarbon, how we can use it, and we'll learn more in the way, right, throughout this webinar. So thank you very much.
Hoping to see you next Thursday.
Mm-hmm.
Thank
Great
you.
talks. Thank you to everybody.
Bye.
Bye.
Hey, Claudia, I couldn't answer your message, but for some
You
reason.
texted me.
I did reply?
Yeah, I got a
Oh, what's up?
I got a WhatsApp, yeah.
Yeah.
Hi,
Because
Oscar.
I try in the chat and it says that you cannot communicate while you're talking or something like that through the chat or something weird.
Okay. Yeah, my Zoom crashed twice. I think I need a new computer. It's the network connectivity on my laptop. It's just horrible. I have a question for both of you before we kick out Oscar, and that's about how long do you want my talk to be on the 17th? And is there anything special that you want me to cover, or should I just talk about my research?
I don't know if maybe Felipe can answer that. I think that half an hour is what
Okay.
we were presenting today. Is good. I think it was a
We're
good
shooting for
time.
30 minutes. I think I couldn't see my clock when I was presenting. I don't know if I actually did more than 30 minutes or not.
Okay.
No,
But
no.
I was good.
Maybe
Okay.
5-6 minutes more.
Okay, so the idea is like I told Sue the same thing. If you pass the time 5 minutes, it's no big deal because we're just gonna move into the questions by the end. So if you extend a little bit, it's okay. And also, you guys are gonna have more time next. You are on the third day, right?
Yeah.
So
September
okay.
17th.
So there's not gonna be an introduction. So we're gonna start sharp at 10:30 Chile time. So maybe 3 minutes late for just let people. Yeah. So
Okay.
I'm
So
gonna
just
start.
anything with radiocarbon in boreal and Arctic, I can string together 2 stories.
Yes.
Excellent. Yeah, no, this is great.
Yeah. No, I think it's cool if you can show. Do you have data for this new probe you have installed in the field? The constant CO2 measurement. Yeah. So that'll be cool. That'll be good to see. Yeah. Yeah. Anything on that, and I think it shows also a different kind of system that works differently. That's a different dynamics, and some people in Chile might get excited about that.
That would be great.
Mm-hmm.
Okay. Yeah, and then I just have some. I